Hello, everyone, and welcome to Lab Manager's Tech Trends webinar series. My name is Mary Beth DiDonna, and I'll be moderating today's discussion, Lentivirus, a versatile tool for cell biology research. We like our webinars to be very interactive, so we encourage you to submit your questions to us at any point during today's webinar. Our speaker will address these questions during the question and answer session following her presentation. To ask a question, simply type your query into the question box, which is located on the right-hand side of your screen. We'll try to address as many questions as possible during our time together. If we happen to run out of time, I'll forward any unanswered questions to today's speaker, and she can respond to you directly if possible. Additional resources for today's presentation are also located on the right-hand side of your screen. I would like to remind you that this webinar recording will be available for free on-demand download shortly following this live presentation, so please watch your emails for information from Lab Manager on how to access this free download. I would especially like to thank BPS Bioscience for sponsoring this event. This support allows Lab Manager to keep these webinars free of charge for our readers. So with that, I would like to introduce our presenter for today's webinar. Veronique Barron, PhD, HDR, is a cellular and molecular biologist with extensive research experience in drug discovery and development, target validation, cell signaling pathways, and cell biology. Dr. Barron has worked in a variety of research environments such as governmental and not-for-profit institutions, biotech, and pharmaceutical companies. She has published numerous patents and scientific manuscripts, has trained scientists at all stages of their careers, and for many years, she sat on grant review panels for the DoD. She is now Scientific Applications Manager at BPS Bioscience. Veronique, thank you for joining us today.
Hi all, thank you Maribeth for your introduction. It is a pleasure to be with you today. I will turn my camera off for the presentation, but I will turn it back on for the questions. All right, so before I present on lentivirus, today I would like to talk very briefly about some of our company's core technologies. BPS Bioscience specializes in recombinant proteins, high purity, off-the-shelf or custom-made with modification or conjugation available on demand. We also offer assay developments with robust biochemical and cell-based assays available in several formats, cell line development for the generation of stable cell lines that express validated proteins or reporter cell lines for cell signaling studies. We have screening and profiling services for custom evaluation of lead compounds to support your drug discovery and development program. And we have an ever-expanding portfolio of lentivirus products with a large selection of derived products of the shell virus particles as well. So today I will present what a lentivirus is. I will talk about several lentivirus systems, how the virus replicates and packages, the PBS lentivirus choices, and I will dive a little deeper into our product line. So I will talk about promoter reporter systems, lentivirus for expression, CAR-T cell therapy, Cas9 lentiviruses, and our latest product line, which is the Pseudotyped lentiviruses for coronavirus studies. So what is a lentivirus? Well, as far as we are concerned, it is a gene delivery tool that is useful for the stable transfection of mammalian cells, and especially ideal for cells that are difficult to transfect. The lentivirus that we use in research are based on the HIV lentivirus. It is a subclass of retrovirus, and therefore it contains a linear single-strand RNA that can integrate into the host genome. So you can see on the left a diagram of a lentivirus structure. You can see that the single-strand RNA is encapsulated into a capsid, and the virus itself is an envelope virus with a dual lipid membrane layer that is decorated with glycoproteins, in the case of HIV, the GP120. And these glycoproteins are responsible for the interaction of the virus with the host cells. And this is how the virus will enter the cell by interacting with a very specific protein on the surface of the host cell. The advantages of using a lentivirus is the large insert size that they can accommodate, up to 10 kb. The fact that the expression is stable because the genome is integrated into the host cell genome with dose effect. These viruses can target a wide variety of cells, and especially they can target dividing or quiescent cells. They can target cells that are immortalized, but also primary cells. And all the primary and the quiescent cells are notably difficult to transfect. So these viruses allow for transfection of cells that would normally be difficult. They are especially interesting because of their high rate of infection that they can reach. Up to 90% of the cells can be transduced after infection with an antivirus. You can achieve very high levels of protein expression if your goal is to overexpress a target protein. And they lend themselves to in vitro cell applications, but also to in vivo applications for those who are interested in animal studies. So there have been several lentivirus systems over the years. The first modification to the HIV lentivirus was to replace the envelope protein with a VSV protein as a recognition motif. So the VSVG binds to the human LDN receptor, and that's a receptor that is present on the surface of many cells. So using the VSV to decorate the virus particle, we can target a broad range of cell types. The first generation had relatively low efficiency, and they were still replicating viruses that required stringent safety precautions. This evolved into a second generation lentivirus system that were optimized for expression and in which non-essential viral elements had been removed. The components necessary for virus replication were split across 3 plasmids, as is shown on the right. And this also requires the use of packaging cells. So the first plasmid is the viral genome, in which we introduce your gene of interest and the resistance gene. And this is under the control of a mammalian promoter. The second plasmid contains the envelope, the gene encoding the envelope protein. And then the third plasmid encodes the proteins that are involved in packaging the virus. So these 3 plasmids are transfected together into a packaging cell, and we most often use the HEK293 cells. Now there's a third generation lentivirus that is also replication deficient. In this system, the virus components have been split across 4 plasmids. Because there are 4 plasmids, the yield is lesser than the second generation viruses, but it is safer. And it is most often used in the clinic either for gene therapy or for ex vivo CAR-T immunotherapy. So at BPS, we use second generation viruses. And this slide illustrates how the virus is packaged and how it can then infect a target cells. So a very important aspect of this packaging is that the first plasmid encodes the viral genome. The second plasmid encodes the envelope proteins that will be expressed at the surface of the virus, and the third plasmid encodes the packaging components. When the viral genome is ready, it is encapsulated by the packaging components, and the virus particle extrudes outside of the cell after merging with the cell plasma membrane. And the virus particle, therefore, contains only the viral genome that contains your sequence of interest, but it doesn't contain anything that encodes for the packaging system. So when this virus infects a target cell, the genome will be processed normally and it will be integrated in the DNA, but this virus cannot replicate and the cell cannot produce any more virus. Because it is replication incompetent, it can be used in a BLS2 biosafety level which is the biosafety level that is found in most cell biology labs. And because it is an invoked virus, it enters the cells with simple membrane fusion. So at BPS, we offer you ready-to-use viral particles offered in multiple sizes. As I mentioned, they do not express viral proteins after infection. Therefore, their replication is incompetent and can be used in a BLS2 biosafety level. We also offer an engineered variant that is integration deficient. And I will come back to this a little later during my talk, but we can offer in some occasions either the integrating and the non-integrating virus. Our latest product line includes pseudotyped spike SARS-CoV-2 viruses that are very useful for the study of coronavirus. So whether you work on CAR T cells, you perform cell signaling experiments or transcription factor studies, you work in immunotherapy, you want a CRISPR-Cas9 system, or you're working on coronavirus, we may have a product that is useful for your research. What are the advantages of buying an off-the-shelf, ready-to-go virus particle? Well, as opposed to buying a cell line, for example, with a ready-to-go virus, you can transfect a variety of cells, and that may allow you to optimize your experimental conditions for the ideal cell line. That may allow you to test different cells to make sure that you're actually working with a cell line that is appropriate for your needs. There is no optimization of virus production. We have done that for you, which can be a huge time saving and cost saving. These allow you to perform feasibility tests, for example, before starting a costly project. You may have a very simple experiment with few conditions that do not warrant setting up a whole virus production system in your lab. And we do quality controls, so you know that you can have reproducible results with our viruses. So these are our product lines. We have viruses that will allow you to study cell signaling, transcription factor activity, and this includes all our promoter-specific reporters. We have expression lentiviruses for overexpression of your protein of interest. Lentiviruses to use in CAR T cell therapy, CRISPR-Cas9 system, and then spike pseudotyped viruses that are useful for coronavirus studies. I will now dive a little deeper into each one of these product lines. So the first one would be the promoter reporter lentiviruses. These are all VSV mediated for infection of a broad range of target cells. We use GFP for green fluorescent protein reporters. We also use luciferase, or you may choose to have a dual reporter system that have both GFP and luciferase. You can see on the right a little diagram for a typical promoter reporter system. You can see that in this case, the reporter gene, which is luciferase, is under the control of a very specific promoter, which is a promoter that detects IL-2 signaling in the cell. So anytime the cell is activated with IL-2, luciferase will be expressed. And you can measure this expression with the luciferase assay. The beauty of this system is that there is a very robust, quantitative, and reproducible measure of the transcription factor activity and the signaling pathway that you are trying to measure. We currently have over 70 reporter systems that includes reporters that measure the activation of AP1, NF-kappa B, STAT3, STAT5, and so forth. And reporter systems that measure signaling induced by IL-6, IL-8, or IL-2, and many more. These allow you to study the activation of a transcription factor, study the activation of a particular signaling pathway. You can determine IC50 for inhibitors, EC50 for effectors. You can screen inhibitors and so forth. So I have represented here two typical results that we can obtain with this quantification system. In the experiment shown on the left, we use force calling to stimulate the CREB transcription factor. And you can see that increasing concentration of force calling activate the reporter system with an EC50 of about 10 micromolar. It is very sensitive and concentration dependent. On the right, we used LPS to stimulate an F-kappa B. And again, you can see a dose-dependent activation of transcription factor and F-kappa B. Our next series of lentiviruses are lentiviruses for expression. So these are also mediated by VSV as the envelope protein. They are all integrating viruses that allow the generation for stable cell lines. High expression levels are reached, and you can study your favorite protein with ready-to-go expression vectors. So ACE2, for example, spike, TMP or SS2, CD32B, and so forth. You can see on the right the schematic of a typical construct for these expression vectors. The ACE2 gene, in this case, is under the control of a mammalian promoter, constitutive promoter, and we also added an antibiotic-resistant gene in C-terminal to make sure that you can generate your stable cell lines. In this case, it is a puromycin selection gene. We have validated every one of these constructs. So you can see on the left, the expression of ACE2 in HEK293 cells following infection with our lentivirus vector. So in purple, we have the control cells, and in green, we have the cells that have been infected with the ACE2 lentivirus. We can see pretty strong overexpression of the protein. On the right, we have generated a stable yellow fluorescent protein reporter primary T cell line after infection with the YFP topaz lentivirus. So you can generate cells that are stably transfected with GFP, for example, or with YAP, or you can generate stable cells that overexpress your protein of interest. Now, we also offer a full CAR T-cell service platform that would include the design and the construction of an antiviral vector, the generation and the validation of functional CAR T-cells, expansion and optimization of the cells, cytotoxicity assay, design of BiTE constructs and production of BiTE antibodies, antibodies screening and development, custom cell line development. So really from the very first step of your project to the very last step of your project, we are here to help you. We can design and perform any test that you need. But we also have now two ready-to-go anti-BCMA and anti-CD19 CAR lentiviruses. So they are both constructed in the same way. You have the anti-BCMA or the anti-CD19 SCVF that is linked to a second generation CAR portion that is intracellular that generates a signaling pathway. So when this construct recognizes the antigen, the signaling pathways inside the CAR T cells are activated, and your CAR T cell can then show cytotoxicity toward your target cell. These can be transfected into various cell types, including primary T cells, and they allow the expression of a functional anti-CD19 or anti-BCMA CAR. The applications are transfection of T cells or Jurkat reproduced cell lines for engineering or for validation. You can study the specificity, the efficacy, or the potency of the construct, and you can evaluate the therapeutic potential of the cells that you're using. On the right are two typical results that can be obtained with this system. So in these experiments, we put together the CAR T cells that have been transduced with the anti-CD19 construct, and we add them to CHO cells, either the controlled CHO cells or CHO cells that overexpress the CD19 of interferon gamma to see if the CAR T cell has been activated. And you can see that when CD19 is overexpressed, the CAR T cell is activated and there is large production of interferon gamma. On the right, we perform the cell type of experiment, but this time we measure the cytotoxicity of the CAR T cell by measuring the killing of the target cells. So in this experiment, the CD19 CAR T cells are added to the target cell. The control is untransduced T cells, and we can see that when the CAR T cells have been transduced with the anti-CD19 CAR construct, they can kill the target cells very efficiently. We then offer a complete CRISPR-Cas9 system. The Cas9 lentiviruses are and can allow for selection of hygromycin or puromycin, and they allow you to achieve stable expression of Cas9 in your cell of choice. Cas9 is tagged with a C-terminal flag to facilitate its detection. Now, when you have obtained your Cas9 positive cells with the lentivirus, you can transfect them with a single guide RNA targeting your gene of interest. And this will allow you to obtain a gene knockout or a gene knock-in and introduce a mutation or add a tag or a reporter to your endogenous gene of choice. On the right, I am showing the flow cytometry analysis that confirms Cas9 expression following infection with the Cas9 lentivirus. So in this experiment, we have used HEK293 cells, but because these lentiviruses do contain the VSV envelope protein on the surface, you can transduce pretty much any type of cell that you want. And these allow you to obtain Cas9 expressing cell lines. Now, we also offer off-the-shelf Cas9 viruses that have been pre-validated to target several genes of interest. So our targets today include PD-L1, PD-1, LAG3, CD4-7, TIGIT, CTLA4, and TCR. But this list is growing, and we can also design and validate for you anything that for your target of interest. So these constructs contain a Cas9 gene along with 4 single guide RNAs targeting your gene of interest. Again, the Cas9 is tagged with a flag tag for easy detection. It will integrate randomly into the genome of your target cell to express both Cas9 and the single guide RNAs, which allow you to achieve very high knockdown efficiencies because they are integrated and the constitutive expression is rather high. So on the right, I am showing a LAG3 overexpressing cell line that was infected with a LAG3 Cas9 antivirus, an integrating virus. And 3 days later, we measure the expression of LAG9 by flow cytometry. And you can see on the left that LAG3 was efficiently knocked down in a subset of the cells. All of these viruses are offered either as a non-integrating or as an integrating virus. And the reason for this is that it can be very, very useful to assess off-target effects. This is unique to BPS. So BPS has engineered a non-integrating lentivirus that is integrase deficient. And on this slide, I'm showing the comparison between an integrating virus and a non-integrating one. So the integrating contains the Y-type integrase. It integrates randomly into the genome. That allows for stable expression of the Cas9 and the single guide RNA, which increases the efficiency of the knockdown considerably. But because it integrates randomly, the integration can lead to off-target effects. Now, in a non-integrating virus in which the integrase is deficient, you can obtain only transient expression of the Cas9 or the single guide RNA, which limits the efficiency of the system. But on the other hand, it eliminates off-target effects. And so if you're using an integrating lentivirus and you obtain very high efficiency of knockdown, but you also are seeing some effects that were not expected, you can use a non-integrating lentivirus to verify that effects that you're seeing are really due to your target, or whether they are due to off-target effects because of the integration. And now, last but not least, we have developed a whole new line of spike pseudoviruses. So in these viruses, the spike protein of SARS-CoV-2 virus has replaced the usual VSV as the envelope protein. So these spike pseudoviruses can infect ACE2 positive cells, and they are very useful to study the infection step of the coronavirus. We have these lentiviruses in reporter systems, so they will allow for expression of the luciferase, EGFP, or both. We also offer dual reporter systems. The spike-mediating infection will drive the expression of your reporter gene for precise and robust quantification of the spike ACE2 interaction in a relevant cellular context. So this is a little diagram that is shown here. We're seeing the virus decorated with a spike protein interact with ACE2. It goes into the cell. The viral genome is integrated into the cell, but in this case, it encodes a reporter protein. Therefore, you can measure the activity of this reporter protein as a direct correlation of the infection. We also offer ACE2 positive cells, HeLa cells, CHO cells, or HEK293 cells that have been transfected to overexpress ACE2. And we also offer Viro E6 cells that overexpress TMPR SS2 as cellular models for these coronavirus studies. The Y-type and variant spike proteins used are envelope. You may need sometimes to use a bold virus as a control to verify that there is no non-specific signal due to your reporter system. But people have also been using our bold viruses to control for non-specific interaction of the virus with the cells and non-specific entry of the virus. So, for example, a mechanical non-specific entries of the virus during infection or something like this. So on the left, I'm showing a diagram of the dual GFP luciferase reporter construct. Again, both the GFP and the luciferase are under the control of a mammalian constitutive reporter. And they will allow the expression of these proteins in your cells. The bold viral particle doesn't contain VSV, or it doesn't contain spike. So technically, it cannot interact specifically with your cell and enter through a normal fusion mechanism. On the right, I'm showing the infection of HEK cells. So on the left, you have normal HEK cells that are not infected either by the bold or pseudo spike pseudovariant. On the right, I'm showing ACE2 overexpressing HEK cells. When these cells are infected with the bold virus, we obtain no signal, showing that there is no non-specific viral entry in these cells. When these ACE2 cells are infected with a spike pseudovirus, however, we see a very, very strong signal that is directly proportional to the entry of the spike virus into these ACE2 cells. So this can be useful to screen neutralizing antibodies against the spike protein, to screen inhibitors of the spike ACE2 interaction, to determine the IC50 of your inhibitor or your antibody, and to examine the effect of variable experimental conditions, for example, pH or temperature of additives on the infectivity of this virus. So I'm showing 3 examples in which we inhibit the infection with an anti-spike antibody or a recombinant ACE2 protein or an anti-ACE2 antibody. So on the left, we have used an anti-spike antibody at increasing doses, and we can see that this antibody inhibits the infection of the cells with an IC50 of 1.3 nanomolar. In the middle, we have used a recombinant ACE2 protein to compete with the interaction of the virus on the ACE2 positive cells. Again, we can see a very nice dose-dependent inhibition of the interaction between spike and ACE2, which prevents the infection with this virus. And then on the right, we have an anti-ACE2 antibody that also inhibits the infection of the spike pseudovirus on the cells. We now offer almost every variant pseudovirus. So as you know, a number of variants have emerged of SARS-CoV-2. The first one was the D614G that was identified in the US. Then there are also variants that have been identified in the UK, in South Africa, and Brazil. All these contain specific mutations in the spike protein, and it is thought that these mutations may impair the recognition of neutralizing antibodies, and they may also impair the efficacy of the new vaccines. So our pseudoviruses will express either the white type spike protein on the envelope, or they will express any one of these variants with the corresponding mutations on the envelope. And it will allow you to study, to perform structure function studies of the spike ACE2 interaction. You can screen new antibodies against the variants. You can compare the effect of a neutralizing antibody against either the white type or the variant. So on the right, I'm showing just one of these comparison experiments. The top panel shows an experiment that was performed with an anti-spike antibody termed clone 4142. We use this antibody to inhibit the infection by the white type virus or to inhibit infection by the B117 virus. And you can see that the antibody is as efficient either with the white type or with the variant virus. On the bottom panel, I'm showing results that have been obtained with 4141 antibody. And in this case, we can see that the efficacy of the antibody is very different between the white type and the variant. With the white type, the virus can totally inhibit in a dose-dependent fashion the infection with the white type virus. However, it cannot inhibit at all infection with the variant virus. So these experiments allow you to test neutralizing antibodies, and they may allow research into new vaccines or into vaccine efficacy. Now, to summarize I will reiterate some of the main advantages of the lentivirus for gene delivery, and that includes infection of a wide range of host cells, infection of non-dividing, as well as primary cells that are notoriously difficult to transfect, obtention of a stable gene transfer, and replication incompetent viruses that allow you to use only a BSL-2 system. We have a variety of products available for research in immuno-oncology, cancer research, coronavirus, neurology, and so forth. We have a wide range of lentivirus-based products, and that includes ready-to-go virus particles, reporter systems, expression cell lines, Cas9 expressing cells, and more. So I would like to thank you all for listening today, and I would be happy to answer questions. I will now turn my camera back on, and we'll take any question.
Okay, great. Thanks very much, Veronique. So at this time, we're going to move into our question and answer session with the audience. So again, for those of you in the audience who may have joined us late in this presentation, you can send in your questions by typing them into the question box, which is located on the right-hand side of your screen. So Veronique, our first question from the audience is, how can I optimize conditions for viral infections when using a new cell line?
Oh, that's a good question. So, well, especially if you're using a lentivirus-based system for the first time, I would recommend optimizing your conditions with a straightforward standard promoter system such as GFP, for example. So if you were to optimize using a GFP, you can use immunofluorescence to detect the fluorescence in your cells that allows you, for example, to perform increasing doses of lentivirus on your cells to see when you achieve maximum efficiency. It allows you to see how strongly the expression that you obtain is. And you can also have some additives, for example, and make sure that your pH conditions, your medium conditions, and so forth are ideal. And so I would really recommend the use of a reporter system to start with, and especially immunofluorescence systems, so that you can visualize exactly what's happening. And then any lentivirus should behave about the same. And so the conditions that you determine with the reporter system should be good for any other lentivirus that you use.
Okay, great. Thank you very much. We have another question that says, are there different efficacies when the virus is packaged in cells other than HEK?
Well, I'm not too sure about this. I know that we use these cells because, you know, they allow for the obtention of very high titers of virus. They're very easy to transfect, and because you have 3 plasmids to transfect, it can be a little tricky with your conditions depending on the cell line that you use. So I can't really answer this question. I don't know how it would work if you were to use other cell lines. It very much depends on the transfection efficiency. You know, the fact that you're using 3 plasmid will decrease that efficiency quite a bit. So if your cell line is sensitive, that may not be very good. And then the high yield of virus that you can obtain.
Okay, wonderful. Thanks so much. I think we have time for one more question, and that is, can your CRISPR-Cas9 system be used in vivo?
Well, in theory, lentiviruses can be injected into animals. At BPS, our products are really designed for research use, and we normally don't produce viruses that are ready to use in animals. So you actually may want to contact us for this, because I think that you need those viruses to be tested for endotoxin, for example. You need to make sure that there's nothing in the medium or in the buffer that would be harmful to the animals. So our viruses are not ready for animal use when we sell them. We can make them ready for you. In theory, lentiviruses can be used in vivo, absolutely. I hope that answers the question.
Okay, wonderful. Thanks so much. So this brings us to the end of today's webinar, and I would just like to remind all of you that this webinar will be available for free on-demand download shortly following the slide presentation. So you can go to the link on your screen, or you can watch for an email from Lab Manager for more information about that. So on behalf of Lab Manager, I would like to thank Dr. Veronique Barron for all the hard work she put into her presentation, and I'd like to thank all of you for taking time out of your busy schedules to join us today. I would also like to thank our sponsor, BPS Bioscience, whose support allows Lab Manager to keep these webinars free of charge for our readers. For more information on all of our upcoming or on-demand webinars, or to learn more about the latest tools and technologies for the laboratory, please visit our website at labmanager.com. We hope you can join us again. Thank you and have a great day.
Thank you very much. Bye-bye. It was nice to be here today.